Proteins are complex molecules that play critical roles in the body. They are composed of amino acids, which are linked in specific sequences to form polypeptide chains. These chains fold into unique three-dimensional structures, determining the protein's function. Understanding protein chemistry is essential for grasping how biological processes are regulated and maintained. Proteins are complex molecules essential for life, composed of amino acids linked in specific sequences. They perform a vast array of functions, including catalyzing metabolic reactions, DNA replication, and transporting molecules. Understanding their structure and function is crucial for insights into biological processes and innovations in biotechnology and medicine. Proteins, derived from the Greek proteos, meaning primary, are crucial, making up about 3/4 of the human body's dry weight. They are primarily composed of carbon, hydrogen, oxygen, and nitrogen, with sulfur and phosphorus as minor elements, underscoring their essential role in biological functions. Amino acids are organic molecules featuring an amino group, carboxyl group, and a unique R side chain. These 20 standard amino acids form proteins, with their sequence dictating protein identity and function. For instance, glycine is the smallest and a chiral, while others possess diverse properties enabling complex functions. Amino acids are defined by their architecture: an alpha carbon bonded to an amino group, a carboxyl group, and a variable R side chain. This R group imparts unique properties to each amino acid, influencing protein structure and function. Understanding this architecture is crucial for grasping how proteins achieve their diverse roles in biological systems. Peptide bonds form through a dehydration reaction between the alpha carboxyl group of one amino acid and the alpha amino group of another. This creates a rigid planar bond CO-NH, essential for protein backbone stability. Polypeptides, chains of 10 to 50 amino acids, form the basis of proteins, which typically contain more than 50 amino acids. Peptide bonds exhibit resonance stabilization, imparting partial double bond character that restricts rotation. This rigidity is crucial for maintaining precise, three-dimensional protein structures, essential for their functions. Additionally, disulfide bonds between cysteine residues provide extra stability, particularly vital in extracellular proteins, enhancing their structural integrity and functional reliability. Amino acids exhibit diverse properties based on their side chains. Nonpolar amino acids like alanine and valine are hydrophobic, influencing protein folding. Polar uncharged amino acids such as serine and threonine form hydrogen bonds. Charged amino acids, including aspartic acid and lysine, are crucial for protein interactions and enzymatic activity. Amino acids are classified by side chain properties. Nonpolar amino acids like alanine and valine are hydrophobic, aiding in protein folding. Polar uncharged amino acids, such as serine and threonine, form hydrogen bonds. Charged amino acids, including acidic aspartic acid and basic lysine, are vital for protein interactions and enzymatic activity. Essential amino acids like leucine, isoleucine, and lysine are vital as they cannot be synthesized by humans and must be obtained through diet. Histidine and arginine are semi-essential, becoming crucial during growth or stress. Deficiencies in these amino acids can lead to health disorders and impaired protein synthesis. Amino acids are categorized by their chemical properties: nonpolar, polar, acidic, and basic. Nonpolar amino acids like glycine and alanine are hydrophobic, while polar ones like serine are hydrophilic. Acidic amino acids, such as aspartic acid, carry a negative charge, whereas basic amino acids like lysine are positively charged. These properties influence protein structure and function. Proteins are complex molecules with distinct structures. Their sequence of amino acids, dictated by genetic code, forms the primary structure. This sequence folds into secondary structures like alpha helices and beta sheets, stabilized by hydrogen bonds. Tertiary structure is the overall three-dimensional shape, while quaternary structure involves multiple polypeptide subunits, forming functional complexes. Proteins have 4 structural levels. Primary structure is the linear amino acid sequence. Secondary structure involves local folding into alpha helices and beta sheets, stabilized by hydrogen bonds. Tertiary structure is the overall three-dimensional shape from side chain interactions. Quaternary structure is the assembly of multiple polypeptide subunits into functional complexes. Protein function is intricately linked to its structure. Precise folding is crucial. Even minor changes can drastically affect activity. Denaturation, caused by heat, pH, or solvents, disrupts structure and function. For instance, enzymes lose catalytic activity when denatured, explaining why fever can be harmful. Proteins are vital for numerous biological roles. Structural proteins like collagen and keratin provide strength and protection. Enzymatic proteins catalyze essential biochemical reactions, while transport proteins like hemoglobin facilitate oxygen delivery. Signaling proteins, including hormones, regulate physiological processes, and defense proteins, such as antibodies, are crucial for immunity. Proteins perform diverse functions. Collagen and keratin provide structural integrity. Enzymes catalyze metabolic reactions. Hemoglobin and membrane proteins facilitate transport. Hormones and receptors manage signaling. Antibodies form the immune defense. Each role is crucial for maintaining biological processes and overall health. Disulfide bonds formed between cysteine residues are vital for protein stability, especially in extracellular environments. They reinforce structures like insulin and ribonuclease, maintaining their active conformation. These covalent bonds ensure proteins remain functional under physiological stress, preventing unfolding and loss of activity. Synthetic biology is transforming medicine with synthetic peptides and recombinant proteins. Genetic engineering advances insulin production, while monoclonal antibodies enhance targeted cancer therapies. Mastery of protein chemistry paves the way for rational drug design, unlocking new biotechnological innovations. Protein folding is a complex process where linear amino acid chains fold into precise three-dimensional structures. These structures form specific active sites crucial for biological functions, influencing everything from enzyme activity to cellular signaling. Understanding this natural origami is vital for advancements in drug design and biotechnology as it determines protein functionality and stability. Proteins, composed of amino acids, are the dynamic architects of life, forming complex molecular machines. They are crucial for health, influencing all life processes and medical understanding. In biotechnology, proteins drive innovations, advancing medicine and technology. Mastery of protein chemistry is key to unlocking new therapeutic possibilities, offering insights into biology and potential life-changing therapies.